Greetings, I bid to all. My name is Sharvin Manickam. I am from USM and today I'll be presenting on my topic for my presentation here, characterizing the overexpression of microRNA 975 in spermatogenesis of Drosophila melanogaster. Thank you. For our first stop, I thought it's best to introduce microRNA right away. So it's a 22 nucleotides long construct. It is brought forth it is synthesized from Drosha and also Dicer in the whole schematics of things that you could see on my right hand side. It microRNA plays a pivotal role in RNA silencing either through translational repression or microRNA decay. Now, for the subject of the hour is microRNA 975. It has been characterized through its overexpression that causes bulging phenotypes in Drosophila. And further down the road, as I'll explain to you much later, it has characterized that it has coiling positive cells, um, coiling positive cells populating the entire bulge of the testis, and my next point of work is to validate those immune genes to see how they respond to it. The wonder bugs, Drosophila melanogaster. Now, the reason why I choose them for my research is because they have a very short life cycle. They have a rapid progeny generation, which means the moment they lay their eggs within 10 days, we can have an adult fly in. They have human organ synonymous functional for about 60%. They have small genome set, and most importantly, they have this UAS GAL4 system. The UAS GAL system is a binary system that is robust for for screening. It is a genetic tool that we use for screening. Now, GAL4 is what we consider to be the driver. The UAS is the expression vector, they are the responder. So one of the parent will have the driver line, the GAL4. And once we mate them and within the progeny, the GAL4 will find the UAS expression vector there and it will overexpress the gene of interest that we will have in that construct. Now, this is the schematic for the Drosophila melanogaster's testis. For the purpose of this discourse, we will be focusing only on the spermatocytes. But however, I'll just give a fly through onto the entire schematics of it. Now, in spermatogonia, we will have the cells rising up from the hub cell, and as they develop into individual spermatids, they will go through the stages of spermatogonia, spermatocytes, and finally spermatids. Now, we use primary antibodies such as fast stream, uh, traffic jam, VASA, and HTS. Now, this tree will specify which of the cells are related to the spermatogonia. Now, once it's into the spermatocyte stage, we will focus only on coiling. And finally, for Bol and Don Juan, as you could see in the final stages, Bol will specify us of the elongating spermatocytes. These are the late spermatocyte before they become individual spermatids. Now, at the terminal epithelium is where we can find those individualized spermatids. Now, my objectives are to characterize the phenotype from by accounting the number of cells during spermatocytogenesis and also the number of coiling positive cells. Besides that, I characterize the fertility rate before amongst the wild type and also microRNA 975. And finally, I would validate the immune response genes arising from the existing sperm transcriptomic data. Now, for the experimental designs, the first will always be husbandry. I will feed them, I will look after them, and once I have a healthy enough population, I will divide the whole group into two. One is to do the fertility test, where I will mate flies, each to either to be a wild type or a mutant fly, which is a 975, to see how the fertility would be. Now, for the second group, I will made them and keep them up for 10 days before I dissect the testes and stain them with those primary and secondary antibodies. And for the analysis, I will analyze for the bulge diameter, the number of coiling positive cells, and also finally for the fertility rate of the fertility test. And after that, for good pictures, for good images, I will use the UV microscopes to get good pictures for the bulge diameter and also the number of coiling positive cells. And as for the transcriptomic data, it will help me for my qPCR validation of the immune response genes. As you could see, the bulge for actin microRNA 975 is definitely more pronounced than the control. While the control have a taper tip, the other does not. The penetrance for actin microRNA 975 is over 700 and definitely more pronounced than the wild type. Now, the bulge diameter for 975 is more pronounced than the control, as you could see here. The coiling positive cells in 975 is once again more pronounced in 975, and as we could see in the pictures in the images much later, these coiling positive cells are shown to fill in the bulge. Now, in this slide, you could see that the coiling is much more concentrated within the bulge when the coiling should have been just only focused in the stalk here. So this is what we have observed when we stain the testes with coiling positive cells. Now, to show the defect in spermatocytogenesis, we stain these testes with bowl. Now, bowl shows the progression of late spermatocytes before it becomes then early spermatids. As you could see, the wavy line that follows over, now that shows the late spermatocytes becoming the spermatids. Now, in the wild type, it is seen to be a typical progression of it. It starts off quite near to the tip before it goes down to the stalk. But here in 975, we could see none of those progression can be seen clearly. But however, the bowl seemed to be focused once again at the bulge, hence concluding the defects in the spermatocytogenesis. As for the fertility test, we could see that despite the bulge, it's very clear that 975 is not as fertile as with the wild type. Hence, in conclusion, the wild type is explicitly more fertile than the 975 flies. As for the discussion, we could have seen that coiling positive cells are concentrated in the bulge as do the bowl. Now I theorize of a defect in spermatocytogenesis and early differentiation. The bowl is shown to fill in the bulge, hence raising the speculation that the cells differentiate early to be spermatids. Some do not even elongate to the stalk. Now, as for my later progression of my work, I will be looking at the suggested upregulated immune-related genes in the transcriptomic pipeline, and finally, I will validate it from through the usage of qPCR. The final verdict is 975 definitely has a defect in spermatogenesis. It is reflected in the fertility rate, and also that the differentiation is seemingly stuck in the coiling positive cells. We postulate that these coiling positive cells are the ones that cause the bulge for its over proliferation. As for my next move, I will be collecting more samples for the fertility test. I will be characterizing the individual spermatids to see the number of it. And finally, I'll be using testis-specific drivers to further elucidate and understand how does this spermatocytogenesis is being affected. the game is afoot.